Last week you learned about fertilization. In this video you are going to learn about the important topic endosperm and its types. Endosperm. Endosperm is formed due to triple fusion. Triple fusion is also called as double fertilization. In double fertilization, among the two male gamete, one of the male gamete is fused with the egg to form zygote and the other male gamete fused with the secondary nucleus to form endosperm. The endosperm is used as a nutritive tissue for the developing embryo. The primary endosperm nucleus is the result of triple fusion, is called as endosperm. So it is used as a nutritive media for the developing embryo. Depending upon the mode of development, the endosperms are classified into three categories: nuclear endosperm, cellular endosperm, and helobial endosperm. So the primary endosperm nucleus divides immediately after fertilization before the zygote starts to divide. The primary endosperm nucleus is the result of the triple fusion. That is, the two polar nucleus is fused with one sperm nucleus to form endosperm. The endosperm carries totally 3n number of chromosomes. The polar nucleus carries diploid nuclei and the one sperm nucleus carries haploid chromosome. So the endosperm is a triploid in nature. It carries 3n number of chromosomes. The endosperm is a nutritive tissue and also a regulatory structure that nourishes the developing embryo. Depending upon its mode of development, it is classified into three categories: nuclear endosperm, cellular endosperm, and helobial endosperm. Nuclear endosperm. The primary endosperm nucleus undergoes mitotic division without the formation of cell wall. The primary endosperm nucleus is divided mitotically to produce two nucleus first without the formation of cell wall. The free nucleus condition is existed in this endosperm, example coccinea, capsella and arachis. So the nuclear endosperm, here the primary endosperm nucleus undergoes mitotic division without the formation of cell wall and the free nuclear condition is existed in the endosperm, example coccinea, capsella and arachis. In cellular endosperm, the primary endosperm nucleus divides into two. Here immediately it forms a cell wall formation between the nucleus. So the repeated division also follows cell wall. So repeated division also follows cell wall formation. Example Helianthus, Ataxia. So in cellular endosperm, the primary endosperm nucleus divides mitotically to produce two nucleus. In between the two nucleus, immediately the cell wall formation takes place and also repeated cell division makes the cell wall formation. Example Helianthus in cellular endosperm. In helobial endosperm, the primary endosperm nucleus moves towards the base of the embryo sac. The primary endosperm nucleus moves towards the base of the embryo sac and divide into two nuclei. The cell wall formation takes place, leading to the formation of a large micropylar and a small chalazial chamber. The nucleus of the micropylar chamber undergoes several free nuclear division, whereas the chalazial chamber may divide or may not divide. Sometimes the chalazial chamber it may divide or may not divide. Example hydrilla and vallisneria. In helobial endosperm, the primary endosperm nucleus undergoes mitotic division to produce a larger micropylar nuclei and a smaller chalazial chamber. The nucleus of the micropylar chamber undergoes several free nuclear division, whereas the chalazial chamber may or may not divide. Example hydrilla and vallisneria. The endosperm may be either completely consumed by the developing embryo or sometimes it may persist in the mature seeds. The seeds without endosperms are called as non-endospermous seeds. Example, groundnut. It is also called as XII seeds. In these plants, the cotyledons is used as a nutritive tissue for the developing embryo. So the some seeds without endosperms are called as non-endospermous or albuminous seeds. In these plants, the embryo is used as cartilage as a nutritive tissue. Example, groundnut and beans. Those seeds which are having endosperms are called as endospermous seeds or albuminous seeds. In this plant, the endosperm is used as a nutritive tissue for the developing embryo. Example, paddy, coconut and castor. Then the last one is ruminate endosperm. In ruminate endosperm, the endosperm with irregularity or unevenness in its surface forms ruminate endosperm. In ruminate endosperm, the endosperm with irregularity and unevenness in its surface forms this endosperm. Example, Araki Katachu. The next functions of endosperm. The endosperm is used as a nutritive tissue for the developing embryo. For the development of embryo, then the endosperm is used as a nutrition. In majority of angiosperm, the zygote divides only after the development of endosperm because the endosperm is used as a nutritive tissue for the development of zygote. So this before maturity of the zygote, the endosperm development takes place. The endosperm regulates the precise mode of embryo development. The embryo development is under the control of the endosperm. So it regulates the precise mode of embryo development. Stages of development of dicot embryo. Example, Caxilla pastoris. It is a crucifer type of plant. The embryo develops at micropylar end of the embryo sac. The embryo develops at the micropylar end of the embryo sac. First, the zygote undergoes the embryo's development is occur at the micropylar end. First, the zygote divides transversely to form or terminal cell and lower or basal cell. The zygote undergoes division to produce two cells, the upper or terminal cell and the basal or lower cell. The further division in the zygote during the development of the embryo leads to the formation of the first stage is a globular stage and it is followed by heart-shaped stage before reaching the mature stage. Before reaching the mature embryo stage, the embryo undergoes certain changes. First it forms globular stage and it undergoes heart stage. Finally, it reaches the mature stage of embryo. The mature embryo has the preliminary root structure is called as radicle and the shoot is called as a plumule and it contains two cotyledon. So it is dicot, so it contains two cotyledon and it possesses hypophysis at this heart stage. The hypophysis is involved in the formation of root cap and the root. The root cap and the root formation takes place with the help of hypophysis and the suspension is used as a umbilical cord-like structure which connects the embryo to the surrounding connective tissue. The use of suspensor is act like an umbilical cord. It connects the embryo to the surrounding tissues. by means of the connective. It functions as the umbilical cord-like structure, and the hypophysis is used in the formation of root and root cap. At the mature stage of embryo, it has a radicle. The radicle is a rudimentary root, and the plumule, it is a rudimentary shoot, and it contains two cotyledon. Topic is seed. The seed is the fertilized ovule, is called as a seed. After fertilization, the ovule is converted into seed. It possesses an embryo, endosperms, and a protective coat. The seed is consist of a embryo and a nutritive tissue endosperm, and it is covered by a protective coat. The seed may be endospermous. That is, if the seed having endosperm is used as a nutritive tissue for the developing embryo, is called as endospermous seed. Example, wheat and maize. In some seed, the endosperm is very small. In this plant, the cotyledon is used as a nutritive tissue for the developing embryo. Such seeds are called as non-endospermous seeds. Example, bean, mango, cucurbits. Then next, the structure of a dicot seed. Example for dicot seed is a sisal, gram seed, a mature mature seeds are attached to the fruit wall by a stalk called funiculus. Mature seed is attached to a fruit wall by a stalk is called as a funiculus. The funiculus disappear and leaving a scar called as hilum. On disappearance of the funiculus, there is a scar is present is called as a hilum. Below the hilum, there is a small pore is called as micropyle, through which the entry of oxygen and water is moves towards the seed during germination. The micropyle facilitate the entry of oxygen and water during the germination. Each seed has a covered by two seed coat. The seed coat is developed from the integument. After fertilization, the integument of the ovule is converted into a seed coat. The outer seed coat is called as a testa, which is very hard in nature, whereas the inner seed coat is called as a tegument, which is thin. The two pea plant, the testa and tegument are fused. The two cotyledons are laterally attached to the embryonal axis or present. The cotyledon stores the reserve food material for the developing embryo in non-endospermous plant. Whereas some seeds like castor, the endosperms contains the reserve food material. Such seeds are called as endospermous seeds. In these plants, the cotyledons are thin. The portion of the embryonal axis are projecting beyond the cotyledon is called as a radicle. The portion of the embryonal axis which are projecting beyond the cotyledon is called as a radicle. It is considered as an embryonic root. It is considered as the embryonic root, or the first formed root system is called as a radicle. The other end of the axis is called as an embryonic shoot, and it is called a plumule. It is the first formed shoot system. The embryonal axis above the level of the cotyledon is called as a epicotyl. The embryonal axis above the level of the cotyledon is called as a epicotyl, whereas the cylindrical region between the level of the cotyledon is called as a hypocotyl. The epicotyl ends with a plumule, whereas the hypocotyl ends in a radicle. The epicotyl is ends in a plumule. The hypocotyl ends in a radicle.